Hello everyone, my name is Nattakorn BUU from Department of Biology, Faculty of Science, BUU University. I am very interested in working on stem cell research. I have a great opportunity to work with Dr. Supalangsi to study how to generate induced pluripotent stem cells or iPS cells from wild cats and domestic cats in order to save wild Felix species from extinctions. We collaborated this work with Zoological Park Organization of Thailand. Let me introduce you to why we need to do these projects. As you already know that many of wild animals are under the risk of extinctions due to human activities. Wild felix are also the great risk of extinction too. In Thailand, there are 9 species of wild cats and some of them are in vulnerable status based on IUCN Red List category, including fishing cat and cow leopard. In order to save these wild felix, iPS cell technology provides an alternative platform to create valuable cells with a great potential to differentiate into all cell types of the body, including germ cells, and even create the whole animal body through cloning technologies. As a model to find the optimal conditions for reprogramming of wild felix, which we have limited resources, we take domestic cat as a model of this research. Here what we have done. We took the cat fibroblasts and then we did cellular reprogramming using cell-replicating RNA expressing Yamanaka factors, including OCT4, SOC2, KRAS4, and c-MYC. Then we got cat iPS cell without modification to its genomes. I also characterized this cat iPS cell by using alkaline phosphatase staining, immunofluorescence, and using QRT-PCR to measure relative gene expression of the cell. Here what we got. We successfully reprogrammed somatic cell, in this case is cat fibroblasts, into cat iPS cell by using cell-replicating RNA approach, similar to what previously done by other laboratories. LIF or leukemia inhibitory factors is required to generate cat iPS cells. The whole reprogramming process took around 18 to 21 days until we get the first appearance of iPS colony. And here, the iPS colony. You can see the bright colony appear in the cultures. We then pick this iPS colony to expand or generate stable iPS cell line. I analyze this colony with immunofluorescence to visualize the expression of some pluripotency marker, including OCT4. OCT4 expression can be found in this clone. We then measure relative gene expression of some pluripotency marker and differentiation markers. And we found that this clone expressed pluripotency markers, but also differentiation markers biased toward mesoderm and endoderms, indicating that some of these clones tend to have spontaneous differentiation. I recently continue to work on the research and aim to finally apply the strategies of this research to wild felix species. So at last, I'd like to thank Dr. Avika Thongvakti and Sandita Karun from Zoological Park Organization of Thailand, and Dr. Supalangsi, my advisor, to give this opportunity to work on this project and be a part of the team to save wild animals from extinctions. Thank you very much.